Bismillahirrahmanirrahim and welcome back to this next video, and this is the 17th video in the series of videos on immunology. And in this particular video I want to focus on the pathogen recognition molecules. If you talk about the pathogens, these pathogens, they express at least a few chemical structures that are not typically found in mammals. And these chemical structures, they are known as the pathogen-associated molecular patterns, or they are also known as the microbe-associated molecular patterns. So these PAMPs and these MAMPs, they are the common foreign structures that characterize the whole group of pathogens. And these PAMPs and MAMPs, they are specific to the pathogens. They are not found on the mammals. If I give you the important example from the PAMPs that you can see on different pathogens, if you talk about the bacteria, there can be a variety of the structure. For example, if you look at this flagellin over here, this flagellin is actually a protein that makes up the bacterial flagella. You can see over here. So this is one of the important PAMPs that is characterized by the immune system of the mammals. We will discuss about that in a while, that how they are recognized, how these PAMPs they are recognized. Another important example, that would be the lipopolysaccharide layer that you only see in the gram-negative bacteria. The third example, that would be the Peptidoglycan layer that you can see both in the gram-positive and in the gram-negative bacteria. If you talk about the yeast cells, there is an important cell wall structure which is known as the zymosine or the zymosine sometime, and that is actually a beta glucan. It is one of the important structures found in the yeast cells. If you talk about the viruses and the bacteria, their nucleic acid is also an important PAMPs that is recognized by the immune cells of the mammals. There are other examples, of course, as well. You can talk about the lipopolysaccharide acid in the bacteria. You can talk about these lipoproteins in the bacteria. There are coat proteins of the viruses, the GPI anchors in the parasites. So all of these, they are the important example from the PAMPs, and these structures are only found in the pathogens and they are not found in the mammalian cells. If there are PAMPs, that means there should be something on the immune cells of the mammal that they are able to recognize these PAMPs. So if you talk about the cells of the immune system of the mammal, they are going to express a variety of the receptors on their surfaces, or in the soluble form you will see that. So these cells of the immune system, they are going to express these variety of the receptor, and they are collectively known as the pattern recognition receptors. For short, we call them as the PRRs. So that means there are PAMPs on the pathogens, there are pattern recognition receptors on the immune cells. These pattern recognition receptors, they are actually proteins, and they are capable of recognizing the PAMPs or molecules that are released by the damaged cells, which are known as the damage-associated molecular patterns, or for short you can call them as the DAMPs. So these PRRs, they have the potential to recognize these PAMPs. They are you can say not good for the health of the mammals, because they are causing certain sort of infections, or if there are damaged cells. Of course, we do not need damaged cells in our body, so these PRR are able to recognize both these PAMPs and these DANTs, thereby clearing them from the mammalian bodies. If you talk about these PRRs, I gave you main examples of the PAMPs. So if you talk about the examples of the PRRs, we usually divide them into four families. The first one, they are known as the toll-like receptors. For short, they are known as the TLRs. And these toll-like receptors, they are very important in the recognition of the proteins, the nucleic acid, and the glycans of the pathogens. That means that these TLR are important in recognizing these PAMPs that are found on the pathogens. Another important class is known as the nucleotide binding oligomerization domain-like receptors. They are known as the NLRs, and they are very important in recognizing the bacterial, viral, parasitic, and the fungal PAMPs. The third type is known as the C-type lectin receptor. So whenever you see this term lectin, that means you're talking about the carbohydrates most of the time. So these CLRs, they are able to recognize the glycans from the wall of the fungi and some bacteria. And the last type, that is known, last family, is known as the RIG1-like receptors, and they are very important in the recognition of the cytoplasmic sensor of the viral RNA. When these PRRs, they detect these PAMPs, whatever the PAMPs is, I gave you many example of that, that actually leads to a cascade of events that label the target pathogen for destruction. So if you look at this is the interaction between the PRRs and the PAMPs that allows the early categorization or profiling of the sort of pathogen of concern. Now this is very important, these interaction between the PRRs and the PAMPs, that is very important because that is going to guide the immune system what sort of immune reaction is needed against that particular pathogen or against that particular PAMP. So you are doing some sort of profiling by the interaction between the PRRs and the PAMPs. Now this profiling of the pathogen of concern, that is very important, as I've told you, because this is going to lead to the subsequent immune response route that will be followed, and therefore the fine tailoring of the immune response as it's developed. I'll give you an example of this. If you remember in my previous video, I told you that we usually classify this pathogen into the intracellular pathogen and the extracellular pathogen. The intracellular being present inside the cell, the extracellular being present outside the cell. So if you talk about the intracellular, for example, if I give you an example to understand the importance of interaction between the PRRs and the PAMPs. So for example, if you talk about the viruses, they are detected via the intracellular receptors that bind the exposed chemical moieties while still inside the host cell. What that means is that if you are recognizing a PAMPs inside the host cell, that means you need specific immune response. So in case of the viruses, if there is an interaction between the PAMPs of the virus and the PRRs of the host cell, PRR in this particular case would be the intracellular receptors. So this can trigger an immediate antiviral response in the infected cells that further blocks the virus replication. This is one of the consequences of interaction between the PRRs of the virus and the PAMPs when you're talking about an intracellular, you can say, pathogen. At the same time, at one hand, an antiviral response is generated. On the other hand, this also leads to the secretion of chemical warning that is sent to the nearby cells that help them guard against the infection. And you can call this as like a neighborhood watch system. I have a detailed videos on the interferons, which is a very important antiviral agent. So once a cell is infected by a virus, it releases the interferons, and the interferon that is working as a neighborhood watch system, that interferon is recognized by the neighboring cell that have not yet been infected by the virus, and the non-infected cell prepare itself to fight against the viral infection that may come along. So I'll continue the discussion in the next video. So if you like the video, please subscribe to my channel, hit the notification bell, do ask me questions, do comment on the video if there is any, and share it with your friends.